Is Dr. Solomon is not here yet.
Yeah, Dr.
Dr.
Bopas and
Salmon?
Dr. Salmon. Yeah.
Dr. Papapas, whatever.
Yeah, I'll just give them a couple minutes. Hopefully they're coming. They responded to the first emails, and then the later ones I didn't get responses, so I don't know.
Normally you would send a reminder, JP.
Yeah, yeah.
Normally you would send a reminder in the morning because everybody is overwhelmed
People
nowadays.
forget. Yeah,
I
yeah.
don't think they would remember what's there on agenda unless it's really something very important to them. I mean, your meeting is part of daily routine.
Yeah, there's a lot going on. Yeah, I was at the main campus to do the TB test stuff, and I saw them giving out the vaccine to people.
Yeah. Hopefully, vaccine will be available to everybody in a few
Yeah, I
months.
wonder when we'll be able to get it.
Mm.
All right. Well, I'll go ahead and get started.
Yeah.
Okay.
So JP, this is a routine required thing, or this is some?
Yeah, I'm required to do this every semester.
Okay. All right. Let's get started then.
Okay.
Can you send email reminder to them now?
Yeah, Dr. Salmon's here. Dr. Bopasa is just not here yet.
Okay.
Can you guys see my screen? Is it good?
Yes, I do.
Okay. So yeah, this is my current model based on the data that I've accumulated in the last since the last committee meeting. Telomere attrition is activating the standard senescence markers P16, P21, P53. Mostly P53, but P16 and P21 are also increased significantly, but not nearly as much as P53. This suppresses PGC1 alpha, which leads to a accumulation of reactive oxygen species. This activates ALCAT1, which reduces the tetralinoleal cardiolipin in the inner mitochondrial membrane, causing impaired mitochondria. And from my recent data, I found that there's a leakage of mitochondrial DNA into the cytoplasm, and there's a huge increase in the cGAS STING pathway, and then an increase in the senescence-associated secretory phenotype in the TERK knockout. In the double ALCAT TERK knockout, all of this still happens. Obviously, the P16, P21, P53 activate, PGC1 alpha, reactive oxygen species are there, but with ALCAT1 inhibited, the tetralinoleal cardiolipin doesn't decrease, and the mitochondrial DNA doesn't leak out into the cytosol. There's a lot less activation of the cGAS and STING pathway, and there's reduced amount of the SASP in the ALCAT-TERK knockout.
JP, can you go to like presentation mode or something? On my screen, your figure is very small, and I'm getting your next slide, last slide.
Oh yeah, let me fix that. Maybe if I put it on this screen. Is it still doing that?
Yeah, can you go down to the presentation, like the play thing? Maybe that'll help.
Maybe I just need to.
Function F5 might work too. Or F8, I think. I don't remember.
Is that still showing the? Is that good?
But it's the same, but it's okay. You can keep going.
Okay,
I don't want-
sorry. I mean, I wonder. Black or unblack slideshow. What if I just do it like this? Is that better?
It's a little better like that.
Okay. So yeah, the hypothesis and significance. So ablation of ALCAT1 improves mitochondrial function by preventing the reduction of tetralinoleal cardiolipin caused by telomere attrition, thereby modifying the inner mitochondrial membrane to be less prone to oxidative damage. This causes a reduction in reactive oxygen species, which leads to a reduction in senescence and the senescence-associated secretory phenotype, thereby improving cardiac function. And the significance of this is a potential therapeutic significance, as this is a druggable enzyme, and the knockout could be mimicked by using an ALCAT1 inhibitor. So I've made a figure outline for the paper. I'm just going to go through that real quick. The data that I've shown previously, I won't spend a lot of time on. I'll focus mostly on the new data that I've accumulated, but just a quick overview. Oh. Disappeared. What the heck? Sorry, 1 second. It's okay. So this was work done at the metabolic core. They measured the O2, the CO2, the heat generated in kilocalories. I measured the systolic and the diastolic blood pressure and the body weight. There was mild improvements in the O2 and the CO2 exchange, mild improvements in the heat, and there was a significant improvement in the systolic blood pressure, and only a slight improvement in the diastolic blood pressure in the males, not in the females, and there was an increase in body weight. So that's going to be my first figure. So then my second figure, I'm going to show the echocardiography data that I collected as well as the sections of the left ventricle that I did H&E staining on. So this is just the ejection fraction. The heart had a lot of significant improvements. The lungs and stuff, not as much, so I'm not going to include that in the paper, but the ejection fraction was improved and the fractional shortening as well. There was no...
JP, I thought that the very first one would be the survival curve. Do you have the survival curve?
Like how much longer they live, or the offspring?
Because the telomere shortening is supposed to eventually kill the knockout mice faster than the double, if our theory is correct. You don't have a record of the survival curve?
No, so I sacrificed them all at 6 months because that's when they start dying.
But that's where you start counting the survival curve. So you would, that's the most important data. If you say a person live longer, but you don't tell how much longer. So that's a very important part of the
Okay,
study.
yeah, they started dying. I don't...
That's what you see. They should start dying. And you will see, hopefully, if your data here presented is correct, the death rate is higher as time go by with the single knockout.
Okay, yeah, I can put that together. It was just a few that died early. I wonder if I could do statistics on it. I'm not sure, but I did record when they died, but that's why I started at 6 months, because that's when everyone started dying. So so then I have the density of the collagen fibers in the H&E staining. This shows up as blue. I just did a ImageJ analysis measuring the amount of blue in each section. There was a significant improvement in the double knockout in male and female. So this is when I start getting into some of my newer data.
I did the survival data that you and Roger were talking about. The Turk knockout mice, what are they dying of primarily?
They have a lot of tumors. One thing that would also kill them, if they got any wounds, like if they got scratched or if they fought or anything like that, they would never heal. And so they just walked around with like a big bloody gash. And that would pretty much be it.
Are they cardiovascular disease or no?
Yeah, so the number one phenotype in the Turk mice is cardiovascular problems, as well as diabetes. Yeah, it's the most common phenotype in this for the Turk model is cardiac dysfunction. My mice specifically, there was a mix. They had, like I said, there was ones with tumors. They had big wound gashes. Yeah, and they definitely have heart dysfunction. That's very apparent. So this is EM from the males and females. The Turk knockout have either missing cristae or destroyed mitochondria all throughout. I did a quantitative analysis where I counted 1,000 mitochondria in each group, and then I did statistics, and yeah, there's a significant improvement in the double knockout relative to the regular Turk. This is max respiration.
What you show is not missing crystal. You're showing some like vacuolation, like white stuff. If you want to show the crystal, you have to enlarge further to see the crystal.
Do another zoom.
Yeah, what you show is not crystal.
Okay.
What you show is some kind of speculation. It's like dead mitochondria left behind the year mark.
Okay. Yeah. Yeah. I should have been
You,
more
you know what the
clear.
crystal look like, right? When they
Yeah.
draw
Yeah. It's the
contour in the
inside
surface,
of the.
yeah.
Yeah.
So on the surface, it's like a wrinkle into the.
Okay. And so I also measured mitochondrial DNA copy number, and I showed this previously. This is the H2O2 generation using the Amplex Red. So I measured the total mitochondrial DNA copy number, the isolated mitochondrial DNA copy number, and then the cytosol mitochondrial DNA copy number. And originally there was no difference in the cytosolic, so I thought that was weird, but it's because there was less total. So I just subtracted the cytosol from the total, and then this is the difference. There's a significant difference between the two once you normalize it for total. So I went back and looked at each generation.
JP,
Yes.
what's your take on that last bit of data? So the transcripts that mitochondria are generating are reduced, but not the cytosol. I mean, how do you interpret that?
So what I think is happening is there's less mitochondrial DNA copy number in general. So it's leaking into the cytosol from the damaged DNA, but because there's less in the first place, there wasn't a difference. And so, like I was saying, if you subtract, if basically I normalize the total to the cytosol, and then there is a difference if you normalize it to the total.
So yeah, I guess my question is the biological aspect, and your thought is that it's because the mitochondria are leaky because they're kind of falling apart essentially with the telomerase knockout.
Yeah. So my belief is it's the suppression of the PGC1 alpha from the p53 activation. So PGC1 alpha is basically it refreshes the mitochondria, and I found that that's suppressed, and so they're not refreshing, and the mitochondria are just basically dying, and it's leaking into the cytosol, activating the cGAS STING pathway. So I went back and I looked at generation by generation, some of the same markers that I looked at in the 4th generation. So this is ALCAT1, P16, P21, P53. Yeah, there's a gradual increase each generation. It lines up pretty close with a P53. P21, I did the densitometric analysis on it, and it does increase. That's my next slide. It's significant. It's not a huge difference, but it is significant in P16 as well. And then that's the same for the males and the females. Females is not quite as consistent, but it does go up. And then I looked at ATM also. So ATM is activated when there's any kind of DNA damage, and so it becomes phosphorylated. So there wasn't any significant difference between the two groups. This male one, I think I should probably repeat. It wasn't very clean, but And then P16 wasn't a huge difference, P21, but P53 had a significant difference, and then CGAS, and then Sting was a huge difference. It just didn't really show up in the wild type and the ALCAT Turk knockout, but it was really bright in the Turk knockout, and that was consistent in both the males and the females. That's what prompted me to look at the
JP, the Sting pathway further downstream, you did not detect it. Sting is very upstream. Further downstream would be the
TPK and that kind of stuff.
Yeah, phosphorylation, TPK, and others.
Yeah.
The reviewer was going to ask you.
Ask me for that. Okay.
Yeah, Sting itself is just the enzyme that bind double-strand DNA. So CGAS actually is the one that binds, and the Sting. This whole thing, it's eventually led to the upregulation of interferon response genes.
Yeah, senescence. Yeah. Yeah. So I also looked at some of the SAS markers. There wasn't a huge fold increase, but there was definitely a difference. And that's just the left ventricle. There's a fold difference, the different groups.
JP, with the P53, is that phosphorylated or whole total?
That's just the total. I was having a hard time getting the phospho to work very well consistently. I do need to go back and look at that again. This is Santa Cruz antibody is crappy. I think I might need to order another one or something, but. And then this is just the quantification of the different markers, the antibodies. So yeah, Sting was super high. And then the ATM obviously didn't change, but the P53 and the CGAS were much higher.
Yeah, but there's not much difference in P21, so that would tell me that phosphorylation of P53 might be the same.
Be the same?
Yeah.
Okay, maybe that's what was going on. Yeah, because it didn't seem to change much, and it was really fuzzy band, but the total change definitely. So I used a couple different antibodies for that, and both times it changed really drastically. So
JP, you also need to indicate which generation of your backcross. This is very important. Is this early generation or late generation?
Oh, this is the 4th generation. This is this right here. This is the, I don't have statistics for this. This is just a single mouse going up. So this is the testes. This was another thing that had the biggest difference when I was isolating them. They were significantly smaller in the Turk knockout, and the ALCAT Turk knockout improved it. I also looked at some of the SAS markers and the senescence Western blots. The ALCAT, I don't know if it's because it just doesn't express as much in the testes or what, but there wasn't really much of a difference. It didn't go up in the testes like it did in the heart. The P16 went up and the P53 is up. P21 did go up, but there wasn't really much of a difference between the two groups.
JP, this is a different tissue. The one you did the Western blot, the different generation from heart. Is that right?
Yeah, this is all the heart. Yeah, this is the left ventricle.
So the testes may have damage much earlier, so there's no need to upregulate anymore. So it's
Mm.
no.
Yeah, I was thinking that too. Like it was just too late. Like maybe if I got it earlier.
You didn't have a different generation, right?
I actually do have the previous generations for
Yeah,
those.
you might want to go back to check the previous
See if
generation.
maybe went up. Oh, that's a good idea. Yeah, I'll do that. And then yeah, there's an increase in the SASP. And this is just a quantification of the Western blot for the testes. So I repeated the female lipidomic data. I was trying to, because the first time I did it, there wasn't a difference in the DHA cardiolipin, and so I repeated it again to see if there'd be a difference. But yeah, there wasn't a difference. There's a difference in the males, the DHA cardiolipin, but in the females there's not. There's a big difference in the tetralinoleal cardiolipin in both the males and the females, but not in the DHA cardiolipin. So the ALCAT1 knockout does restore the tetralinoleal cardiolipin to close to normal levels, but the DHA cardiolipin was exclusive to males and not females.
This is only in the heart, right?
Only in the heart, yeah.
And the apparently the rescue in female is more significant than male. Which one has the double knockout? Which one has the
male or female?
Yeah.
I think it was pretty close across the board.
Okay.
Didn't really see a huge difference. And then I did the testes lipidomics, and the tetralinoleal cardiolipin was increased in the ALCAT-TERC knockout, similar to the heart.
You went through the very beginning very fast. I remember you did the telomere length measurement.
Oh yeah.
That should be the first thing the reviewer asks you. Well, you should
Yeah.
present this in the beginning.
In the beginning.
Yeah, this
Okay.
is where all the bread butter about.
Yeah, this is it. It didn't change the telomere length.
Ideally, you want to show this side by side with the survival curve. Unfortunately, you already zapped the mice. This is what, if I'm a reviewer, I would say, does it prevent the telomere shortening and extend lifespan? That's the two most important questions.
Okay. Yeah. Yeah, lifespan would have been good. Yeah. Okay, and then my seventh figure is just the model. So this is kind of just going to be a summary. Just wrap it all up. And then yeah, that's going to be it. My seven figures.
JP, could you? I mean,
Sorry,
I'm having a hard time figuring out the in part because I think because you're including multiple tissue systems, which I can understand in some ways. But can you sort of sum up what the paper says in a few sentences, how you view it?
Yeah. So just going back to my model, basically, it's improving the ablation of ALCAT1 is improving the inner mitochondrial membrane, and that's reducing senescence and improving mitochondrial function, basically. And that's this is a whole body knockout, and the telomeres is also, the TERC is a whole body knockout as well. So it's having kind of a global effect. on the mouse, because it's not like cardiac specific or anything like that. So
What's your rationalization going to be for including heart and function and testes / ovaries? You know, that's a harder reach for me. You know, as a reviewer, I'm be like, why did you pick these two systems? You know, on one hand, you could say, I want to do all the systems, right? I want to look at heart, I want to look at muscle, look at brain, whatever.
yeah,
On the other, you could say, I only want to do one or a very few selected related. I mean, these are, you know, kind of, you know, hand waving. They're not really related, right? I mean, I wouldn't group reproductive function with heart function. So, what, you know, what's your thought on how you're gonna
well,
drive those?
that's a good question. So the heart and the testes are the two things that are affected the most by the telomere shortening. If you look at the Turk model, it's mostly heart, diabetes, testes, ovaries. Obviously, and infertility is the biggest problem. They start to go infertile at the 4th generation. So yeah, that was going to be how I was going to justify it. That there's some parts of the mouse that it doesn't really affect very much.
I mean, it may not be a problem. It seems kind of disparate at this point, right? I mean, you're getting findings that are relatively similar between the two, which is, you know, makes good reason to do it, right? If you said, okay, look, it protects all these heart things, doesn't do anything in the testes, I'd go, you know, so what? They're really different systems, right? So
Right. Yeah, they're not connected like the heart and the
Yeah.
kidneys or whatever. Yeah.
You know, I think it's something you, as you're writing it, you're gonna want to keep in mind. I think Rogers mentioned this too. You know, as a reviewer, what's a reviewer gonna point out, right? Where's the flaw? What is it that or not a flaw? Where's the concern that I have? Why did JP do this study? You know, that's what you're gonna have to think about.
Yeah. Okay. Yeah, I actually did like all the H&E staining, and I did Westerns, and I did a bunch of stuff on the lungs, but there wasn't really any difference. And so because I did that once I got the respirometry data back from the metabolic core. I was like, oh, there's huge difference in the O2 and the CO2, but yeah, I couldn't find much of a difference, so I moved on.
Yeah, I mean, you might want. I don't know, because I don't have the paper in front of me. You know, a version, so it's hard. But you might want to think about: do you want to include lung data to show? Listen, we looked at other tissues. These are the primary ones where it seems to be affecting it.
Exclusive to those two, yeah. Okay.
Just, you know, keep it in mind. I wouldn't probably put stuff like that in the actual text. It would be more of a supplemental thing.
Supplemental thing. Okay. Yeah. Well, yeah, that's good advice, because then I could just put it all in, because I collected it all, and it is interesting that it's exclusive to different parts of the body. Okay. And yeah, as far as future studies, I've said this before, but yeah, our lab has ALCAT1 inhibitor that they use on different models. Obviously, can't use it on these at this point, but using an ALCAT1 inhibitor on some tissue specific, either Turk or different senescence models, I think would be important. Further investigate the function of ALCAT1. Why does it cause an increase in reactive oxygen species? It seems to consistently increase inflammation, and so I'm thinking it's part of the immune response, and I think that definitely needs to be looked at further. And then investigate if ALCAT1 contributes to senescence and other models of aging, whether it be like the TERT or like the doxorubicin models.
For the point of your second point there, have you or the lab looked at immune function in ALCAT knockout mice or the function of immune cells?
So one thing that's interesting is the cells are really hard to infect. If you overexpress ALCAT1, they're basically like immune to any form of infection. So we try and like put in like green fluorescent protein and stuff like that, and it just blocks it out because the inflammation just goes through the roof. But as far as specifically looking at it, looking for it, no, that is something I think that should be looked at though.
I think JP, that's not correct. I think Dr. Salmon asked the right question. Right now we have at least 2 people in the lab focusing on the role of ALCAT and inflammation. Remember all the work on ARP3?
Oh, NLRP3. Yeah, inflammation.
Yeah.
But I mean, as far as like
People like,
resistance to disease and stuff like that.
Yeah, we're focusing extensively right now on the role of ALCAT and chronic inflammation. Remember the tissue-specific knockout of ALCAT from adipocyte, and also using the imaging mice from GDKPC.
Oh, the P16 mice. Yeah.
All very clear-cut data that ALCAT regulates cellular senescence and chronic inflammation by multiple mechanism. So that's.
Yeah, I mean, it's chronic inflammation. Do you think the chronic inflammation is like a low grade like heart defense? Like it's defending against
every week, right? We discussed the significance of chronic inflammation in aging-related diseases. They are there everywhere, from diabetes to cardiovascular to neurodegenerative. So, and one important feature is ALCAT function as a prion-like protein.
That's really interesting.
Aggregation of A beta and tau. Those are really new frontier for the field.
Yeah, the prion stuff is super interesting. The brain stuff. So yeah, does that answer your question, Adam? Yeah, as far as like what it does, the positive role, I do think it needs to, like, why would it be there? Like Dr. Shi, do you think the chronic inflammation is just like preventing like inflammation or preventing infection of the heart, like endocarditis or something like that? Can you hear me? Oh no.
You're mute.
Am I muted?
No. Roger's
Oh, Dr. Shi's muted. Oh yeah.
Are you asking me? I thought you asked Dr. Salmon.
Oh no, I was asking you, like, what would be the point of the chronic inflammation?
Question, you know, why inflammation in your age-tert mice is so important? How that connected with the aging and the deterioration of aging-related phenotype you're seeing? Because this is, once again, the reviewer will ask you, is this how ALCAT improve the aging-related phenotype? Just by attenuating inflammation, prevent a STING pathway activation, or even that activation, you didn't do it. What else is involved?
Yeah.
So that's where you need to think about ALCAT to do multiple things. So the STING pathway is just part of it, and there's mitochondrial dysfunction. I'm not sure you measure the mitochondria change in your double knockout versus single. Mitochondrial respiration.
Oh yeah. I did I isolated the mitochondria and did the seahorse. It's in one of the one of the figures. Yeah, it's
So basically, you need to put just like Dr. Salmon asked you in a sentence to, very short paragraph or a couple of sentences, describe why should the reviewer care about this story? What's the central hypothesis, and what's the evidence you prove this central hypothesis hold water? What you show me was only the PGC1 alpha part, but I believe there's more than that.
Look further. Okay.
Because you have a cellular senescence. Did you check the literature that PGC1 alpha regulates cellular senescence?
Yeah, that's part of the they call it the axis of aging, this P53 PGC1 alpha mitochondrial pathway. It's been pretty extensively looked at, especially with telomere shortening. It's
But in your drawing, if you go back to the last slide, it did not really elaborate how this SASP, how that's related to your aging phenotype.
So CGAS and STING have been pretty heavily implicated in senescence as well, from what I've read. It's pretty common indicator, because it's the DNA and the mitochondrial DNA are leaking into the cytosol, and so it's basically signaling the cell to stop, to enter into a senescent state, and that releases the SASP.
So JP, did you look at SASP concentrations? I don't remember from the top.
Yeah, I did. I did some PCR here.
Okay.
These are some of the more common ones.
There's
Judas
not much
Campisi.
difference.
Yeah, not a huge difference. There was a couple fold difference here and there, but this is fold difference. But yeah, it's not like 6 or 7 fold, but
Well, for IL-6, there's almost no difference. Oh, wait. There is. Okay.
yeah. So
see.
the red is the red, and the brown are the
Okay. Yeah. So there is a difference.
yeah. MMP3 wasn't very different, but it's the matrix metalloprotein. It's kind of seals the cardiomyocytes together. Anyway, yeah, that's it. Any other comments or questions?
JP, you have a target journal you're looking for this manuscript?
Yeah, I was looking at either Aging Cell. I noticed there's a Nature Aging that just came out. I don't know if that would be good or not. It's like a brand new journal, but I was
Cut out for a second.
Nature Aging, like a new nature journal that just came out not too long ago. So I thought about doing that, but yeah, I think Aging Cell would be appropriate for it. Do you have any recommendations?
No, I don't have any recommendations. I mean, kind of have to see what the paper looks like in the
final
end.
thing. Yeah. I'll send it to you. I have the basic outline. I'm gonna put in this mitochondrial DNA data that I just collected recently, but yeah, I've actually written the manuscript too, but I'm gonna continue improving on it a little bit. Molecular metabolism's been really nice to me, so I thought about maybe when I published a paper with them and they put me on the cover of for the month. It was really cool. It's unrelated, but
JP, this is too good to be
molecular metabolism.
Yeah, I would still try second tier I mean, we are not a celebrity lab. We are not the club member. If we are club member, this would be Cell Nature Science paper. I don't dream about it. It's a lot of politics involved, but at least you should try a second tier, like a PNS, cell metabolism. I'm not sure it would pay much attention to aging study, but even aging cell is a bit low because the scope is too narrow just for aging.
Hmm.
So I think those are the backup journal. You should
PNAS
find
or
a
something.
better journal first.
Go a little higher first. Okay.
Because the telomere shortening is still a commonly recognized model of aging, and
Yeah, everybody's familiar with it. So
so how you slow down aging from telomere shortening has no clue. I don't think anybody come out any close about it. I mean, this is not reverse. This is probably prevention. There's no one can reverse it.
yeah, it's mostly just attenuation, but it's definitely significant for most things. So especially with the senescent stuff, it's pretty cool. Okay. Well, thanks everybody. I sent the forms, so just the second and third page. You just need to rate me, and if I need it today, if you can. So just sometime today, just fill it out and send it back to me.
Okay, bye.
Okay. Thanks
Great.
everybody.
Bye, everybody.
Bye.
Happy holiday.